Hello everyone, welcome back to part 3, the final part of episode 9, the visual system. Well, this time I don't have an eye in my hand because now we're at the farthest place from our eye, the cortex. To be more specific, the primary visual cortex. We're going to start with that. We're going to go from the primary visual cortex to higher cortical areas. And then, after we finish looking at the entire visual system, we will be able to take a look at some visual impairments. The primary visual cortex. The primary visual cortex is numbered the Brodmann area 17. It is located in the occipital lobe at the back of our head. The primary visual cortex has some part at the surface of the back and some part in the middle, in the midline of our brain. In the primary visual cortex, a very important feature we observe is a conserved retinotopy. We've mentioned in the first part of this episode that topy means topology. The topology, or relative locations of things on the retina, are preserved in the cortex. There are certain changes that the information has undergone. For example, the left visual field is now on the right primary visual cortex, which we abbreviate V1. The right visual field is on the left V1. The upper visual field is now on the lower part of V1, below a fissure called the calcarine fissure. And the lower visual field is now in the upper V1 above the calcarine fissure. In terms of the vertical structure of the primary visual cortex, as usual, as all neocortices, it has 6 layers in terms of lamination. The 6 layers are from the outermost to the innermost, 1 through 6. Layer 4 is further divided into 4A, 4B, 4C alpha, and 4C beta. These cortexes are usually divided into columns. Obviously, columns are vertical. This is because most connections in the cortex are radial. They are perpendicular to the surface of the cortex, rather than tangential. Let's take a look at the detailed connections between the thalamus and the cortex. Magnocellular LGN cells mainly project to 4C beta neurons. Parvocellular ones usually project to 4C alpha neurons. Coniocellular cells project to layers 1 and 3. It also projects to a region known as the blob. Inside the cortex, 4C alpha neurons project to 4B neurons, and 4C beta neurons mainly project to 3 neurons. In these layers, there are pyramidal cells that send axons out of the cortex to other regions in the cortex. They go down from the gray matter on the surface of the cortex into the white matter just beneath it. They travel along the white matter and go to other regions in the cortex. The information flow from the eyes is shown through the ocular dominance columns. We have already seen this picture in episode 8.3, which is about the activity-dependent wiring of the brain during development. The ocular dominance columns are mainly dominated by one eye. That is to say, the input from one eye is preferred to the input from the other eye. There are some binocular regions in between the ocular dominance columns that are innervated by both eyes. Because of these dominance columns, V1 is also known as the striate cortex, meaning that there are stripes here. Visual information processing. Binocular vision is an important feature that is processed in the primary visual cortex. There is also a selectivity to information that is passed down. For example, we have orientation selectivity. This means that the neurons are sensitive to a specific orientation in the visual field. For example, perpendicular to the ground or horizontal. Some cells are very sensitive to lines that are oriented like this. Or maybe we have a cell that is very sensitive to lines that are 42 degrees from the horizontal. We can see in this figure that different orientations are colored differently. The different colors represent columns of neurons that are sensitive to different orientations. We also have direction selectivity. The direction here does not mean the direction of a line, but rather the direction of movement of a line. For example, moving from the left to the right or moving from up to down. These selectivities are achieved through certain cellular mechanisms. For example, simple cells are selective to orientation. Let's say a cortical neuron, a simple cell, has input from 5 LGN neurons, and these are the 5 receptive fields of the LGN neurons. As we can see, they are lined up. Therefore, when a horizontal line of light is shown on these receptive fields, then the LGN neurons will be activated to a greatest extent. Then the LGN neurons will be activated to a greatest extent, and the cortical simple cell will be excited to the greatest extent. Complex cells have more complex receptive fields and are able to process more features, including orientation, direction, and much more. There are some regions in the primary visual cortex called blobs. We've mentioned them before because they receive input from the corneal cellular LGN cells. They're called blobs because when we stain the cortex for a protein called cytochrome oxidase, these blobs stand out. They express a very large amount of cytochrome oxidase. Scientists used to think that they have some distinguishing properties from areas between the blobs, also known as interblobs. However, blobs and interblobs, as we've later discovered, probably do not have such distinguishing properties. We're still looking for the difference between blobs and interblobs in terms of function. From what we've talked about before, we can construct a model called the cortical module. This is because if we take a small column of around 2 mm times 2 mm from the cortex, then this small column, or cortical module, can present a lot of information from the visual cortex. Firstly, it will include both the right eye dominance column and the left eye dominance column, as well as a binocular region between them. Second, it will contain some blobs. And thirdly, it will have orientation columns of all orientations from 0 degree to 360 degree. Therefore, we say that this cortical module is representative of a certain region in the visual field. Okay, we now see this map again. We have successfully exited the eye, gone through the thalamus in the LGN, and reached the first region in the cortex, V1. Now it's question time. Please try to define the following terms. A. Retinotopy. B. Ocular dominance columns. C. Cortical module. Here are the answers. Retinotopy means the spatial mapping of visual input from the retina onto the LGN, then onto V1, preserving relative positions. B. Ocular dominance columns means column stripes in V1 where neurons respond preferentially to input from one eye over the other. And C. Cortical module means a functional unit of V1 containing all orientations and both eye dominances, as well as some blob areas. Time for higher cortical areas. We will first go from V1 to V2 to V3. V1, V2, and V3 are all mainly located in the occipital lobe. V2 is around the borders of V1. As you can see, V1 is yellow and V2 is orange here. V3 is further around the borders of V2. V3 is represented in blue here. V2 may be responsible for illusory contours. For example, do you see a star here? I believe most of us would see a star, but actually our brain is being fooled. There's no star here. There are only 5 Pac-Mans and 5 angles here. The illusory contour of a star may be created by V2. In the further areas, we can divide these areas by 2 streams, the dorsal stream and the ventral stream. The dorsal stream, as its name suggests, goes dorsally to the parietal lobe. It solves the problem of where. It deals with motion. The ventral stream goes to the temporal lobe. It solves the problem of what. It helps us recognize things. First, the dorsal stream. The dorsal stream has a component called V5. It senses motion. For example, it has cells that are sensitive to a very narrow range of directions of movement. Its cells usually have large receptive fields and are able to sense motion across a large area. MST, which stands for medial superior temporal area, is an area that is also selective for motion. There are cells that are selective for linear motion, going like this, for circular motion, going like this, and radial motion, going like this. The MST is important for navigation in space, for motor perception, and for controlling our eye movements. Now in the ventral stream, we have V4, which is important for color perception, the inferotemporal IT area, which has output to learning and memory areas. The communication to learning and memory areas is very important because when we see something, the only way that we can recognize it is by remembering it. A specific area in IT is called the fusiform face area. This area is quite peculiar. Some neurons respond to specific faces. For example, one neuron might respond to all faces that are related to Star Wars. For example, one neuron may be specifically respondent to an actor in the film The Lord of the Rings. It may also have a weak response to other characters in the film or to the actor in other films. That is to say, this area is a general recognition area for faces. An area beside the IT is the visual word form area, which is simply an area that helps us recognize the form of words. So it is surely activated as you are watching this video and looking at the slides. The process from sense to perception is quite complex. We have not yet understood them fully, but here are some factors that contribute to this transition. Firstly, from the retina to the thalamus to the cortexes, we have gradually larger and more complex receptive fields. This helps us deal with more complex information. We also have a process and integration of various types of visual information as we go along the pathways. And finally, we combine context of stimulus, expectation, attention, memories or prior experiences all together to form our perception of a sensory stimulus. This is the complete map of the visual pathway. Question time. Please choose the one single correct statement. A. The earliest cortical visual areas are located in the parietal lobe. B. Area fusiform face area is located in the frontal lobe. C. The dorsal stream is in charge of recognition. D. There are neurons in area IT that respond to faces specifically. Please take a moment to pause and think about it. The correct answer is D. For A, the earliest cortical visual areas, which is the primary visual area, V1, is located in the occipital lobe. B, area fusiform face area is located in the temporal lobe. In fact, the fusiform gyrus is where this area is located, and this gyrus is inferior in the temporal lobe. C, the dorsal stream is in charge of processing movements and motions, while the ventral stream is in charge of recognition. And D is correct. There are neurons in areas IT, more specifically in the area FFA, that respond to faces specifically. Okay, that ends our tour of the visual system. It's time to get to some visual impairments. There are various degrees of visual impairments. A person could be completely blind. If they are completely blind, they might still have a sense of light and dark, or they might not. A person could simply have impaired vision. This could include loss of visual field and/or loss of visual acuity. Loss of visual field means that we can't see that much. For example, we only have central vision, or we only have peripheral vision, unable to see the center, but able to see the periphery. Loss of visual acuity means that we can't see things very clearly. For example, very severe myopia is an instance of loss of vision acuity. There are some other cases where there are loss of specific visual functions, such as the perception of color. Visual impairments also come from various causes. For example, physical damage, as in accidents or tumors. Genetic diseases such as retinitis pigmentosa, color blindness, etc. Other diseases such as glaucoma or diabetes. And impairments can come at various loci. It can impact the eye, the photoreceptor, the retina, the optic nerve, the cortex, etc. That is to say, it can happen in almost any part of the visual pathway. Let's go to these loci one by one. In the eye, we've talked about refractive errors including myopia, hyperopia, presbyopia, and astigmatism. Myopia is short-sightedness, hyperopia is farsightedness, presbyopia is a senior condition, and astigmatism is where we can't focus light on the fovea. We also have strabismus, which we talked about in episode 8, part 3. This includes wall-eyed, where the eye are unable to focus and are all looking outward, and cockeyed, where both eyes are looking inward and unable to focus. And cataract. Cataract is the condition where the lens becomes opaque. This usually happens in elderly people, where beta-crystalline and other proteins in the lens become tangled together and become opaque, no longer transparent. In the photoreceptors, we have a disease called retinitis pigmentosa. It is usually a rod dystrophy, where the rod photoreceptors die out. As they die out, first, there is a loss of nocturnal vision. This has a fancy name, nyctalopia. Then there is gradual loss of peripheral vision, as rod cells are the main type of photoreceptors in the periphery. And finally, there is a loss of central vision, where cone cells die out together with rod cells. Retinitis pigmentosa is sometimes a congenital condition caused by gene and is sometimes due to some mutations. Color blindness. Color blindness come in many varieties. We have deuteranomaly, protanomaly, tritonomaly, and achromatopsia. Deuteranomaly is a kind of color blindness where we don't have M cells. M cells, as we may recall, are cone photoreceptors that are responsible for receiving green light. Protanomaly is the case where we have a deficiency of L cells, which are cone cells that are responsible for red light. Deuteranomaly and protanomaly are both cases of red-green color blindness. In fact, the genes that code for M cells and L cells are very, very close in evolutionary relationship. They are also very, very close on the chromosome. In fact, both are on the X chromosome. That is also why boys are more likely to get these color blindnesses than girls, because girls have 2 X chromosomes and boys have only 1. Since this is usually a recessive gene, boys are more likely to get this color blindness. And tritonomaly is a case where there's S cell deficiency, a lack of blue cone receptors. This is rather rare. And achromatopsia is a case where all cone receptors are non-existent. Retina-related impairments. A macular hole is the case where the macula is entirely destroyed, sometimes by physical damage. In this case, the entire central vision will be lost. Branch, hemicentral, or central vein occlusion would cause blood to be occluded in the veins, and this will damage the retina where this occlusion happens. Diabetic retinopathy is induced by diabetes. Too much blood sugar in the blood will cause damage to the retina. Also, age-related macular degeneration is a degeneration of the macula as age advances. This is usually manifest as a gradual loss of central vision. Stargardt disease. Stargardt disease is a congenital disease caused by a mutation of the ABCA4 gene. It causes an accumulation of toxic byproducts in the retinal pigmental epithelium, which is just outside the retinal photoreceptor layer. These toxic byproducts will harm the retinal pigmental epithelial cells as well as the photoreceptors. Optic nerve-related impairments. The most famous one is glaucoma. Glaucoma is the case where, well, let's leave that for a little bit later. Let's first take a look at this. In the anterior chamber of the eye, which is between the lens and the cornea, there's a fluid called the aqueous humor. The aqueous humor is usually leaked out in a little duct at the edge of the cornea, so it does not accumulate in the anterior chamber. However, if this little duct is blocked, then the aqueous humor will not be able to escape from this anterior chamber. It will accumulate and cause the pressure inside the eye to go up. This is the case of a closed-angle glaucoma. There are also open-angle glaucomas where the duct is not blocked, but nevertheless, the intraocular pressure is increased. This pressure compresses the optic disc, which is the place that the optic nerve leaves the eye. This damages some of the axons in the optic nerve and gradually causes loss of vision. Another optic nerve-related impairment is the pituitary tumor. The pituitary gland is very close to the optic chiasm. If the pituitary tumor grows too large, it will compress the optic chiasm and cause the axons to function abnormally. Cortex-related impairments. Physical damage to the cortex, for example, a shock to the occiput to the back of your head, will of course hurt your cortex. There are also some higher-level cognitive cortex-related impairments. For example, if you have damage to the parietal lobe, sometimes you will have trouble recognizing half of your visual field. We will put them later when we talk about higher cognitive functions. That's all for visual impairments. A final question time. Please discuss the clinical implications of these cases. That is to say, will there be a disease? Case A. A man has a malfunctioning OPN1-MW gene, which codes for M cone cells on his X chromosome. B. A person has a pituitary tumor that is very large. C. A child has strabismus, but the condition was diagnosed and corrected when he was 1 year old. And D. Both maculae of a person were burnt by laser. Here are the diagnoses. A malfunctioning OPN1-MW gene in the only X chromosome will be called deutanopia or deutanomaly. This is a type of red-green color blindness. B. A big pituitary tumor will cause loss of peripheral vision on both the left and the right because the optic chiasm is compressed. C. Corrected strabismus is likely to have no problems. That is because the strabismus was corrected at an early age and the brain is still plastic enough. It has a lot of plasticity to correct the problem and regain normal vision. D. Burnt maculae will cause macular holes. This leads to loss of central vision but intact peripheral vision. In this episode, we went to the primary visual cortex, talked about further processing of visual information in higher cortical areas, and discussed some visual impairments. That's all for our trip in the visual system. In the next episode, we're going to enter the auditory system. See you again in episode 10.